about unsupervised learning image segmentation.
Okay, thank you. I'm Dan Guo, a PhD student in computer science from Northeastern University. So the title of my presentation is Unsupervised Segmentation. Unsupervised segmentation of mass spectrometric ion images characterizes morphology of tissues. So I'm going to start from introducing the background of mass spectrometry imaging and current data analysis. So different from LC-MS, mass spectrometry imaging can characterize spatial distribution of ions throughout the sample. So to illustrate how MSR works, here I'm showing a very interesting example, a painting of a farmhouse, and thanks to Dr. Graham Cooks for sharing this data with us. So why this data is interesting? Because we can see different colors from the painting, and different colors have different chemical compositions, and mass spectrometry can tell us which chemical or which molecules in this color. So in MSI, a pixel is the location on a sample. And then we can obtain one mass spectrum for one pixel. So one mass spectrum for one pixel. And the experiment goes through every pixel in this painting. And then we have a lot of spectrums for a lot of pixels. And if you take the values, the ion intensity values of some MRZ, and we encode the ion intensity in colors, and we can plot this in figures. And so this is called ion images. So what is single ion image? It's a color representation of ion intensities of just one MRZ across pixels. So most current methods are using all MRZ as the input of their model. So the main idea of this work is to analyze one MRZ, one sample at a time. So it has different analysis goals in MSI. So one of them is clustering. So there are two different kinds of clustering. One is clustering pixels, or we see image segmentation, and another one is clustering MRZ. So the goal of clustering pixels is to find segments of similar chemical compositions using the information of all MRZ. Or more importantly, we'd like to find MRZ with intensity selective in some subregions. And similar, the goal of clustering pixel is to find groups of MRZ with similar spatial distributions. Because pixels in an image is spatially correlated, so when we do clustering pixels, it's better to incorporate the spatial dependence of pixels. So spatial K-means segmentation was proposed in 2011 ISMB. It's the first one to incorporate spatial dependence when clustering pixels. And then spatial centroid K-means was proposed, and it also adds lasso regularization to the model. So as we can see from the results, so it's interesting because it's like the segmentation based on the information of ions. And we can see, you can see the patterns can be mapped back to the original painting. But it cannot find all segments. So by segment, I mean colors in the original painting. Like we don't see the differences between color brown and color light green. That is one of the limitations. So how to select region selective ions is that we can calculate a Pearson correlation coefficient between an ion image and a region of interest. So the region of interest can be defined by one segment from the segmentation method, or can be defined by some optical images. So different ions have different spatial distributions, as we see from the previous slides. So if we just do clustering based on all the MLVZ, we have one segmentation map. It cannot represent different spatial patterns of different ions. So another goal in MSI is class comparison. So unlike clustering, so for class comparison, it analyzes one MRVZ at a time. So for the painting data, we have just one replicate. So what if we have many replicates for different classes, like in this mouse brain data? So thanks to Dr. Linkoff and Dr. Laskin for sharing this mouse brain data with us. So in this data, we have two classes, and we have multiple samples for these two classes. And we want to know whether one specific MRVZ is differentially abundant between the two classes or not. So what should we do? So the normal method is to compare the average ion intensity across the tissue between two groups or two classes, and then put the average ion intensity to statistical methods like t-test, ANOVA, or linear regression. So it's very obvious that if we just average ion intensity across the whole tissue, then we kind of lose the spatial information. So we lose the spatial heterogeneity. So it does not take any advantages of the MSI at all, because we don't see the spatial patterns. So next I'm gonna talk about the proposed model, spatial DGMM. So first, let's look at the motivation. So here, the motivation is to find a spatial representation of each ion image, which can be used to all the downstream analysis goals we just talked about. So first of all, we need the representation to preserve the spatial information. And second, we need this representation to be ion-specific, which means different ion will have its own representation. So the intuition that we model the ion intensities are from different subgroups, and for each subgroup, the ion intensity follow a specific distribution. And also we can locate pixels from different subgroups, like these pixels are from subgroup 1, and these pixels are from subgroup 2. In order to do this, we propose spatial DGMM. Spatial DGMM is a dirichlet Gaussian mixer model with spatial dependence. So we assume that there are key subgroups per ion image, and the ion intensity of pixels are from a key Gaussian component with unique means and variances. So the component membership of a pixel is following dirichlet process, and the component membership of pixels are spatially correlated. So here is a graphic representation of the model. So xi is ion intensity as pixel I. So once again, so we analyze one MRVZ at a time, one sample at a time, and we repeat for different MRVZs. Then K is the number of Gaussian components. Mu and sigma are the mean and variance of components. So mu j, k are the number of parameters of interest. So alpha is the duration prior, and pi is the probability of pixel I belonging to component j. Pi is dependent on alpha, and z is a label of pixel. So z is derived from pi, and x is dependent on z, mu, and sigma. So the posterior probability of location I in component j is defined in this formula. So to impose spatial smoothing, we defined a weighted linear combination of posterior probability of the neighboring locations, and we add this we add the linear combination of the posterior probability to the dirichlet distribution, and then we derive pi like in this formula, and we have the negative log likelihood like this, and we use expectation maximization algorithm to get the parameter estimation. And once again, mu, j, k are the parameters we are interested and will be used as input of downstream analysis. Okay, so next I'm going to talk about the results part, including evaluation of the model and its application its applications in downstream analysis. So like there are some parameters we are interested in. So to evaluate the model performance, we need to know whether the model can correctly predict the, correctly estimate the parameters. So here we simulated some eye images with ground truth. So here in this eye image, we have 3 known Gaussian components. Component 1 is the circle, and component 2 is background, and component 3 is the triangle. And each of them has its own mean. So we simulated the eye intensity in this formula, and so we added spatial term phi to this eye intensity. So the spatial term is modeled by ICAR model, intrinsic conditional autoregressive model. So here are some results. So from the so for low noise level data, so we can see it's quite clean, and both Gaussian mixture model and spatial DGMM can have low mean intensity estimation error and low component misclassification rate. And when the noise gets higher, the Gaussian mixture model can be very noisy, but still spatial DGMM can still have lower mean intensity estimation error and lower component misclassification rate. So within the noise of 20%, the model can perform very well. So after that, we applied our model to real data, experimental data, and the first experimental data is AAS mouse spring data, which was obtained from a multi-MSI experiment. So this experiment was done by Dr. Catherine Rollins. So as we can see from the result of spatial centroid segmentation and spatial k-means segmentation, so it has very noisy segments. So it does not infer any segments related to the tissue morphology. However, we can see some MOVCs have very structured spatial distribution, which is somehow related to tissue morphology. So in this case, spatial DGMM can uncover these ions and can summarize different spatial distribution patterns of these different MLVC. So the second experimental data is CPG and silane preconditioned mouse spring, which was obtained from DAISY MSA experiment. So, and this one is the same data set we talked about in the introduction part of class comparison. So this time, spatial centroid k-mean and spatial k-mean segmentation can refer some segment which can be potentially mapped back to the tissue morphology. However, once again, so this cannot represent the individual spatial distribution patterns of individual ions. So in the following, I'm going to talk about how this model can be applied to downstream analysis and the advantage of using this model instead of just the input of eye intensities. So as we talked about in the introduction, so one of the goals is to select ion with a selective distribution in some specific regions in a tissue. So the commonly used method is Pearson correlation coefficient. And here we show this example that Pearson correlation coefficient can actually fail when the spatial distribution patterns are very complicated. So here the gray area is a predefined region of interest, and we want to find some ions with intensity selective in this region. So for MOVC 844.67, so it has a low Pearson correlation coefficient, but we can see from the eye image it has this morphology of the predefined region of interest. But if we fit this eye image using spatial DGMM and match each Gaussian component to the region of interest and select the highest matched component, we can see the gray we can see the gray component actually is a good match. So here we show that spatial DGMM can model complicated spatial patterns where Pearson correlation coefficient can actually fail. So another important goal is to cluster MLVZ of similar spatial distribution patterns, because this helps us to understand the biological process and the relationship between MLVZs. So, and hierarchical clustering is commonly used for this task. And here we want to compare using intensity or using Gaussian component as input of the hierarchical clustering. So we simulated 40 ion images, and they have 4 different spatial distribution patterns, and 3 of them are heterogeneous distribution, and 1 of them are homogeneous distribution. So if we do hierarchical clustering on the intensity space, we can see from the clustering result. So MLVZ clustered in one cluster are actually not always from the same spatial distribution pattern. However, if we do hierarchical clustering on the Gaussian component space, so we can see it's very close to the ground truth, the simulation data here. So another task is class comparison. So here we show the advantage of using spatial DGMM instead of using average ion intensity across tissue. So again, the mouse spring data, we have 2 classes, CPG preconditioned one and silent preconditioned one. And we want to know MLVZ 826.76 is differentially abundant between the 2 groups or not. So we can take the average ion intensity across the tissue and compare the average ion intensity here, and we can see from the result, so the CPG group has higher average ion intensity than the silent group. Or we can fit spatial DGMM first, and then we have 3 Gaussian components per tissue, and we have their means and variance, and then we can compare the mean of Gaussian component between 2 groups. And we can see that actually for component 3, the CPG group has lower intensity than the silent group. Except for absolute intensity, you can also compare relative intensity like here. So it's kind of doing normalization on the whole image. And we can see the difference between the 2 classes are larger in the relative intensity. So to conclude, so we propose spatial DGMM, which segments an ion image into a data-driven number of Gaussian components. So our simulation shows that spatial DGMM accurately estimates the Gaussian components. And spatial DGMM uncovers ion-specific patterns which cannot be found by multivariate analysis. And so using the results of spatial DGMM as input to downstream analysis improves the accuracy. So, and spatial DGMM is implemented in Cardinal. So you can visit this website for more details. And last, I'd like to thank Dr. Olga Vitek's lab, and thanks to Dr. Lenkov, Dr. Laskin, Dr. Cooks for sharing their data. Thanks to ISCB for their travel fellowship, and thanks to NSF for their funding. Thank you.
Okay, question for Dan? I have one if nobody else does. So I'm not quite sure I followed how the spatial smoothing was done, but it seemed like it was, as a regularizer, it seems like you should be able to formulate that as a prior as opposed to a sort of a posterior smoothing step. Have you considered formulating it that way?
Oh, actually, it's formulated as a prior because we add the linear combination in the Dirichlet distribution. So Dirichlet is a prior of the.
So the spatial smoothing is in.
Yeah, right.
Great. Any other quick questions? Okay, I think we need to move along anyway. Thank you very
Okay.
much, Dan.